So hello everyone, thank you to the organizers for having thought of me on this beautiful morning. So today I'll be presenting some of the final work that I carried out while I was at the Technical University of Munich, where we worked on the effect of seed dormancy, so seed banking, on the evolution of populations, and also on how to detect and to infer seed banks. Now, in a lot of the inference models, we ignore selective forces because when we want to infer things from genomic data, we generally concentrate on neutral processes. But we know that these can have effects, so it's important to understand how much selection will affect the genome structure in general. Okay? So we started with a simple question, which was to try and understand how positive selection will affect genomic signatures, or signatures in the genome. So first of all, I'm going to start with a slightly long introduction because I'm a biologist and we have to justify what we do. So seed dormancy or seed banking is what we call a bet hedging strategy. So what it does is that individuals will produce descendants that will go, some of them will go into a dormant state and they'll be saved in the seed bank. And others will germinate immediately and contribute to the next generation of reproductive adults. Now, with time, bit by bit, individuals from the seed bank will also end up germinating and rejoining the population. Now, this strategy is said to have two advantages. One that's a demographic advantage, which is very obvious. If we're in an environment that is very perturbed, demographically speaking, if one generation, no individuals reproduce, well, we still have individuals in our seed bank that can germinate or even hatch if we're talking about an egg bank, and create new reproductive individuals to reproduce. Another advantage is that we have a sort of temporal storage of genetic diversity. And this is something that's been talked about for a long time because what happens is with these seed banks, rare alleles can be maintained for a longer time in the population because they're not immediately being lost in the reproductive population through genetic drift. And just to be precise, genetic drift is not the mathematical drift, it is actually the stochastic component of the diffusion process, okay? So in biology, whenever we talk about drift, we mean the stochastic process. Now, one of the consequences of these seed banks is that we end up with a rescaling of the generation time. And in general, this is rescaled by 1/b, where b is the germination rate. So if in general within a population, the probability of germination or becoming an adult is 0.2 per generation, while the generation time is 5. Okay, very simple. Now, another way of seeing this is how this rescales the coalescent. So here on this side, we have a classical Wright-Fisher coalescent. We have 4 individuals. And we easily see that the time to the most recent common ancestor is 4n, okay? So it's 8 steps as we go from one side of the tree to the other. If now we have a population in which there is a seed bank, even though we have 4 adult individuals in the seed bank, the fact that we have these difference in germination rates and individuals can stay dormant for several generations in the soil, or eggs stay dormant, individuals no longer select their parents from the generation right before them, but they can select them from much further back in the past, okay? And this instinctively will increase our coalescent time. And this by a rate of 1/b squared, okay? Still the germination rate. And from this, we can infer our effective population size, which is what we often use as a description of to quantify genetic drift. It is actually greatly increased by this b squared because we divide n by something that's between 0 and 1, okay? Always the germination rate is between 0 and 1, with 1 being the Wright-Fisher model. Now, in previous works where we wanted to understand how these seed banks would affect the dynamics of selection, Koopman et al. Proposed a diffusion model. I'm sorry, is the graph? Yeah, that's fine. Proposed a diffusion model in which they could estimate the times to fixation of a beneficial allele, a single beneficial allele at a locus under selection. Now, from this model, they weren't able to infer the probabilities of fixation, but we're going to get to that. And what they found was, here we have the relative times to fixation. So we took the Wright-Fisher time to fixation as our reference, okay? And here we have the length of the seed bank, which is basically 1/b, so the generation time. And we see for the neutral case, which is in this dotted line here, so sigma is equal to 0, as the number of generations increases, we definitely have a scaling that is exactly as NE is. So it's scaled by 1/b squared, okay? So we have the time to fixation that follows the expected Kingman coalescent. However, in the presence of selection, we are no longer following a scaling that follows how NE has been changed, so how the neutral diversity has been changed. And we end up in a scaling process that is much shorter in time. It only depends on B. Okay. The consequence that this has is that even though in general we see that the time to fixation is longer, so it's a slower process. However, because the scaling is not by NE, it actually means that selection is acting in a more efficient way in these populations. So selection is stronger relative to drift. Now, so this brings me to the first question I'm going to be trying to answer today, which is, okay, so we have these times to fixation. We weren't able, through the Koopman approach of the diffusion process, to estimate probabilities of fixation. And the question remains, though, okay, we know how the times are. What about the probabilities, and how can we measure that? So this will be, yeah?
So I'm just to know whether I understand correctly. So when you say that selection is stronger compared to drift, but in a sense, the seed bank allows you to recreate individual types that would have disappeared through selection. So what does that mean to compare the two? You know, I mean, selection would mean that you're able to purge some alleles, but then they reappear because of the seed bank. So what do you mean by this?
Well, I think the way you can look at it is when we look at the classical diffusion approaches, one example is like the Caballero and Hill approaches, where they looked at NE that was changed, the effective population size that was changed by selfing. The probability of fixation of a rare allele was still scaled by NE. Whereas in this case, it's not the case at all with the seed bank. We're no longer scaled by NE. So selection is not happening on the same scale as drift is. Whereas in the case of selfing, for example, selection is occurring on the same scale as NE. And it's not the most intuitive thing to understand. I completely understand you were confused. But yeah, so there's something happening that we're not completely able to explain just by using the effective population size as we usually do as a scaling factor. So something else is happening. So my first question will be how this affects probability of fixation. And then the second part of my talk, which will all be together, it's one simulation program, I swear. I'm not going to go further than that. The second question that we want to ask was, okay, we know that the seed banks affects the coalescence, but we also know that it rescales genomic processes. And that means that it will change how a selective sweep or a selective event will affect the rest of the genome. That's what I was talking about in the beginning. And what this does is that it actually rescales the effective recombination rate, which is given by rho, and the effective mutation rate, which is given by theta. So rho is a function of the actual genomic recombination rate, but scaled by NE. And theta is also the actual mutation rate scaled by NE. So just to give you a quick example, I keep wanting to point with my hand, but I can't. So just to give you an example, if we look at the Wright-Fisher model here, so we have no seed bank. We'll look at the case without recombination, so the effective recombination rate is 0. And we have a theta of 1, so this is the effective mutation rate. We see in this case, we only have, for example, 3 alleles that are maintained, or 3 SNPs that are maintained in the population. If now we move to a population with a seed bank, just B of 0.5, which means the generation time is now 2 and no longer just 1, we move to a stronger diversity already of 12 different alleles that are maintained. And this is simply because of the fact that theta now is equal to 4 because of this rescaling by B squared. Okay? So we automatically accumulate more mutations just through this process. If on top of that we add recombination, we see that with the, in the case of no seed bank, when rho is equal to 1, in the case with seed bank, it is scaled by b, 1 over b, so it's equal to 2. And we see that completely changes the topology of the trees, and so we need to take these things into account. Okay? This is, it'll be clearer when I start showing the graphs with the genomes, so you can have a clearer idea of how this works. Now, because of this rescaled genomic processes, it has been suggested, so this is just a verbal model, which we're going to be testing today, that if we look in a population, if we look at in a population with a seed bank and without a seed bank, and we have one locus that's right here given by this red trait that's under selection, we expect that because of the increased recombination on either side of this site under selection, and because of the increased mutation rate as well in the seed bank compared to populations without seed banks, that we will actually end up with a much stronger decrease of genetic diversity right around the locus under selection, okay? And that we should be able to observe a much narrower and stronger decrease in diversity right around that area, more so than in a classical population. And potentially that could mean better detection of such sites, okay? Now, so this is the second part of my talk, which is just to understand how selective sweeps, how they are, how they look, and if they follow the existing predictions. So it's a very simple simulation model. It's only simulations. We have a single isolated population, diploid individuals. Population size is fixed at size n. There's random mating. Populations above ground are discrete. So they only live for one generation, they produce only once. We have a fixed germination rate, B, over time, and the mutation and recombination rates are fixed. One strong hypothesis that we make in this kind of model is that mutation continues even during the dormant phase. Now, there are reasons to accept this and to refute this assumption. But there is empirical evidence that supports, because of the underlying metabolic activity that's still happening within the dormant individuals, there are mutations that are still accumulating. So it seems to be the case that there are actually mutations happening nonetheless. Now, when we sample the parents, what we do is, in this very complicated C++ code, I'm not going to get into it, what we do is that we have our current generation is produced by individuals that are sampled first by their age. So we say, okay, what age group am I taking from? And once I've chosen the age group of the age of the seed or the individual, I will then select within that group, age group, the individual with the best fitness. Okay? And this is how reproduction happens every generation. And once these individuals are produced, they have a certain probability, 1 minus b, of going back into dormancy and then joining the seed bank, and b of germinating and joining the next generation. Okay. So the simulations are pretty simple. What we do is, let me just draw this on the, because I forgot to include a slide on this. So what we have basically is we have our genome. That's right here. We have an infinite number of sites. Okay. And each generation, what happens is that we have a mutation rate that's defined to introduce new neutral mutations throughout the genome. They can be anywhere, so the distribution is uniform of where these mutations happen. We also have a recombination rate that is also uniform throughout the genome. Okay, so we can end up with mutations and recombination events happening. And in order to simulate the selective sweep, we have a single locus. Okay, so here it's 2 because we have 2. So let's say big A is the wild type and little a is the selective mutant. And this is just at a single locus. And what we do is we introduce a single mutant, a single allele that's under positive selection, and we follow its dynamics. Okay. So at the start of the simulations, what we do is that we have 100,000 generations of burn-in time to make sure that throughout the genome, there's no positive selection at all in the beginning. Throughout the genome, we arrive to a mutation drift equilibrium in the population. Once that happens, we introduce the single copy that has a fixed selective force. And then we follow the allele frequencies throughout the genome, and we actually keep all of the genealogy using a very complex tool called TS-Kit. And that will help us keep track of everything that is happening everywhere in the genome. Okay? Now, just as a reminder, this is what we're trying to see if we can end up with this kind of pattern. So, here we are at the results. First of all, we just wanted to check with the neutral case that we really do have the same expected results with our simulation model compared to expectations from classical models with seed banks. And what we see here, going from no seed bank to a seed bank generation so 4 generations of seed bank, you can ignore the effect of the recombination rate for now. I'm not going to be talking about that today. We see that, yes, this does follow the pattern of a rescaling by NE, okay? That's just it. So we know that the model works. There's no problem. We've taken we calculated the NE over all of the genome, and we come to something that is coherent. Now, when we looked at the times to fixation, contrary to previous results, we find that depending on the effect of selection, so here each line represents a different effect of selection, the neutral fixation time is actually hidden under here. When selection is very small, so almost neutral, selection is scaled by b squared, just like the NE is. As soon as selection increases, so here if we look at these lines of 1 and 10, they're right here, we change the selection regime, and now we're scaled by b. The reasons behind this aren't clear. We've been trying to figure this out, but we don't know why this happens, but something happens at the dynamic level. Now, when we look at the probabilities of fixation, we go from top to bottom, no seed bank. Sorry for the figure, I chose the wrong one. 2 generations of seed bank. I think this was 4, no, what is this? 3 and 4 generations of seed bank. This is an expectation for the fixation probability of mutations under strong selection. Fixation probability, this is our selection coefficient. And what we see is that the more we have dormancy, the lower the probability of fixation. So we actually end up losing the allele more often with a seed bank than without. And this result for us was extremely counterintuitive. It was actually slightly disappointing because it goes against all of the ecological theory where we were saying, hey, look, seed banks, great. We can adapt because we're keeping all this genetic diversity. But that's not actually what's happening. And just a small reference to the people in the room, we figured out what's probably happening. So this is the seminal work by Champagnat, Ferrier, and Meillard that showed that there are 3 phases in the probability, in the trajectory of an allele. We have the first phase that is completely stochastic here. Once it passes a threshold, it follows a completely deterministic trajectory, and then it ends up at another threshold, where there it becomes stochastic again before it goes to fixation. And what we realized when we were looking at these simulations is that with the seed bank, we spend much more time in this first phase. So either it's possible that the gamma threshold is much, much higher with dormancy than without dormancy. And that means that it's very rare that the allele frequency actually passes this threshold and joins the deterministic trajectory to go to fixation. So counterintuitive result, but it was actually good to understand why that was happening. Now, when we wanted to look at the signatures of the sweep, so this is our neutral genetic diversity with seed bank and without a seed bank. Okay, so blue is with and black is without. And what we see without rescaling, just the brute results, is that yes, first of all, we do have a lot more diversity, nuclear diversity. This is the site of selection, and yes, we have a very steep selective sweep that's happening. But when we rescale the diversity, so we divide by the mean diversity observed in the Wright-Fisher model, we realize that yes, it's narrower, but it's not necessarily much steeper when we're looking with seed banks than without seed banks. So this is something that might be problematic when we're trying to detect our sweeps. Now, just to show that this is not a simple scaling of NE, this is the same result as previously. So we have the selective sweep happening here. Here, so this is a seed bank of a B of 0.35. Here we compared different Wright-Fisher models, but with different effective population sizes. So here's 8,000, 2,000, 1,000, and sorry, there's an extra line there. And we see that despite rescaling the NE to fit the expected effective population size with a B of 0.35, we never get the same dynamics. So everything that's happening with the selective process and the genomic process is not simply a scale of NE. More is happening. Okay? And this has to do with the recombination that's been rescaled and the mutation that's been rescaled and everything that's underlying that. So just to my last results slide, what we wanted to do was, okay, what happens now if we want to detect these sweeps? So we used a tool called Omega Plus that takes genomic windows, looks at the diversity there, and sees if something out of the norm happens in that specific region, and says, okay, here there's a probability that there's a sweep. And it gives a sort of threshold value. So below these lines, we can detect the sweep, and above them, we can no longer detect them, okay? And we did this for the case with, so here there's no sweep in the full line here. And this is after a certain number of generations with 2,000 generations being all the way at the bottom after there's been fixation. And we see in general, when there's no seed bank, we're much better at detecting the sweep. And this is, again, because of the fact that before it was extremely narrow, so it actually means that we have a tougher time finding it. And when we look, and here we have, should be smaller window, sorry, not larger window. And when we look at the case with the seed bank, so B is equal to 3.5, when we take the default window for this tool, we find that we actually almost never detect the sweep. We never find it, despite the fact that it's narrower and should be steeper. And by making it a smaller window, so instead of taking big chunks of the genome and looking if something strange happened, we take much smaller sizes. It slightly improves, well, it greatly improves it, but not as much, it's not as easy to find these sweeps as it would be without a seed bank. So this can have, of course, ecological implications. If we're working in a species that does have seed dormancy, we might never be able to find sweeps that we're looking for, selective sweeps that we're looking for. So as a quick perspective, my first question was, is there a lower adaptive potential in species with seed banks in this case? Well, not necessarily. Just because there are lower probabilities of fixation, it does not mean that in general there's lower adaptive potential. And this is mostly due to the fact that we're making some limiting assumptions, which we have to make at some point, which is, first of all, the environment does not change, which means selection does not change. So maybe that is part of what would play a role in the adaptive potential of the species of the seed bank, and we don't have this at all. And also, we're looking at a single locus and a single allele under selection. And this might also be limiting. Why? Well, because first of all, it's possible that we have several advantageous alleles that are segregating with potentially the same selective effect, and those will all be maintained, or several of them can be maintained within the population. And that is something that we do need to test further. And it would seem that there are actually more soft sweeps in populations with seed banks, and that's in the paper that was just submitted at the population genetics group at the TOM. So maybe we're looking more at soft sweeps than strong sweeps in populations with seed dormancy. So with that, I would like to thank you. And in white, you have Kevin and Aurélien, who are the co-authors on this paper. And if you have any questions.
Thank you, Diala. So are there any questions, comments?
Thank you very much for the very nice presentation. So I'm very happy that you raised this perspective because it was exactly my question. What can you do with a fluctuating environment? Because if I understand well, this is one of the motivations for having this sort of model to explain the advantages of seed dormancy, to have a repeated environment with different, even with only one positive selection at one selection at only one locus.
I mean, this could be realistic for traits that are always under strong selection, right? So there are some very basic metabolic traits which will always be under strong selection no matter what the environment. So for those, we will never see these selective sweeps. It'll be too complicated. But yes, I do think that if we do have this fluctuating selection and we end up with balancing selection, the fact that we have a seed bank will definitely be advantageous. But that's the next step that we're going to have to implement, hopefully, soon.
Thank you.
Thank you.
Thank you, Diala, for your talk. It's a comment more than a question. So I just would like to mention the probabilistic works by the, we could say, Berliner School around Rohrenblatt. And they have many papers working on the seed bank effect and in a probabilistic point of view, including adaptive dynamics case. Perhaps you could have a look, and also population genetics models.
But the Johann Blatt models are a very different kind of seed bank because they consider a very long seed bank, which has a completely different dynamic. So the fact that their dormant individuals have an exponential distribution in their ages brings us in a very different context. Here we have a limit as to the time individuals can spend in the seed bank, which corresponds more to plants and to animals and vertebrates, whereas in the Johann Blatt models, they're more in the bacterial realm. So, and we've noticed when we've compared our models that we do not have the same results. And that's due to the fact that they have these exponentially distributed age groups. You're welcome.
Thanks for the. So I was just wondering about your fixation probability results. So if one sort of thinks dimensionally, then if your NE scales as 1 by b square, and since selection must scale as 1 by time, so essentially as b, so then NS, so is it then surprising because NES scales as 1 by b, so one would imagine then that the fixation probability is lower with a seed bank, or?
So if we have selection that will be scaling by b,
because if selection has units of 1 by time, which now.
So yeah, selection will not be as strongly scaled as NE. I agree with you, but this is the thing though. With these kinds of models, what we've realized is just from the time to fixation, is that we can't really use NE as a reference anymore. So I mean, I understand where you're coming from, but I don't see how. But maybe we should talk about this. Maybe there's a way to look at this closer. Peut-être.
Yeah, but your talk was so great that there are too many questions.
Thank you.
Now, I would just like to further second the comments by Vincent. So when you think of fluctuating environments and fluctuating selection, you have to deal with time scales as well. And so if I know NE is not the right reference to compare, but in a sense, if your seed bank lasts longer or allows alleles to remain longer than your changes, you will not see the same pattern as if changes are takes much more time than your seed banks allows. Yeah. You have different regimes.
So if the changes take longer than the age of the seed bank,
then you will essentially have a constant environment for the time your seed come back to the real population.
I should know the answer to that. I should have
okay. I don't know. My comment was it depends a lot on how frequently.
It will definitely. I think if it's too frequent, it might not be good either. So I mean, you have to have something that's in the middle, and usually. So what I know is from the timescales with host parasite dynamics, with the seed bank, there you really see something that it goes into a cycle that will depend on the age of the seed bank. But there you have two things that are controlling each other. When you have something that's independent of this, I mean, definitely it shouldn't be too long. Otherwise, there's no reason to seed bank. But I think if it's too short, there wouldn't be a reason to seed bank either. So it'll be, yeah, you're right. It'll have to be intermediate frequency. It's something to test.
So thank you. We can thank again Diala for this night.
Thank you.